Antonio, for this introduction about the seminar. I want just to say some words about the CV of David Perahia. David was one of the pioneers in using quantum chemistry to understand biological molecules in the group of Pullman at the early 70s. He has published a lot of paper about that. I was very impressed. At this time, people work not a lot in these biomolecular systems. And David was one of the first one to give a lot of contribution there. After, he has also a collaboration with Professor Karplus. That was the Nobel Prize in 2030. And he was working about the normal modes. He has developed a lot of things about the normal modes. And he has also pioneered and reference in these methods. And finally, maybe the most important for us is that we have started a collaboration between France and Brazil in 2010, about 2010, and we have published a lot of work after that, and many people was exchanged. Mauricio is one example of that. He is there, and we are very proud to collaborate with David, that was one of the pioneers in molecular modeling, in biophysics, in biology, molecular biology. And so let's see what's new that he was doing now. And so let's see the conference of David. Thank you, David, to accept to talk for us this night.
Thank you very much. And I am very pleased to participate to this conference series. And from Brazilian Biophysical Society. And I thank you very much. I'm very happy to talk here, and thank greatly to the organizers for inviting me to present our work. Then I will share my screen. Yeah. Okay.
It's okay.
It's okay?
Yes, it's okay. Oh, now it's not okay.
Now it's not okay?
No, maybe.
It's okay now?
Yes.
Okay. Now I will be presenting a new approach for atomic flexible fitting of macromolecules in Cryo-EM Maps using molecular dynamics and normal modes. And I will be sharing this presentation with Mauricio Costa, who is in postdoctoral stay now for the third year, I think, in Paris. And he will take part in this presentation for presenting the new method that we developed with MDeNM-EMfit. And then after Mauricio Costa, Charline Fagnan, who did her PhD in our laboratories and then applied the MDeNM, the method that we developed. She applied to a membrane protein, which is called KIR, and this also in relation with electron microscopy. And then all this work were done in two laboratories. One of the laboratories is headed by Catherine Vignan-Briand, who is professor at the University Pierre et Marie Curie. And she is involved in biophysical characterization of macromolecules and also their functional properties. And myself, I am mostly involved in molecular simulation. And our two laboratories, then I am from École Normale, I'm in École Normale Supérieure Paris-Saclay, France. And our two laboratories, the one from Professor Vignan-Briand is at the historical Quartier Latin, which is the intellectual center of Paris. And my laboratory, we shifted in 2020 to a new region. We were near Paris, but we are now in Saclay region. You see the building here is brand new building, but I was there for 2 or 3 weeks because of the pandemic. And but the region is very, very rich. There are many institutions all around. There are the Cyclotron Soleil, and there are the Atomic Center studies. There are École Polytechnique. There are the University of Paris South, and many, many other institutions. But we didn't have yet the chance to work there. Maybe it will be possible in 2021. Now, what I wanted to say that electron microscopy was a very powerful technique which started many decades ago. And you see in the blue, I think this is a very revolutionary technique because we can see the structure of biological molecules that cannot crystallize and which are in the solution or can be embedded in membranes or not. And we can see the large complexes and very, very nice method. And you see that in blue that many structures are increasing from 2000 at the beginning of the century. Now we reached, if you cumulate all the structure, we reach 8,000 structures which were resolved. And the other side, you see the other curve, in orange curve, the number of the resolution is decreasing. And you see, and in the beginning of the century, it was almost 9 Armstrong. And now in the best cases, we are reaching 1 Armstrong. But even all the biological macromolecules are in the range resolution between 3 and 4 Armstrong or more. But this range of resolution is already sufficient to have good modeling and see what are their structure. And all this curve comes from the electron microscopy data bank. I think this is very revolutionary because now we can visualize the structure of macromolecules in their functional state in the real environment, which was not the case with the X-ray crystallography. I think all these trends I remember in 1980s. We see the same trend, the number of crystal structure and now we see the same thing with EM electron microscopy technique. And I think it was no surprise that the Nobel Prize were attributed to the pioneers in this field. Now, all these things were feasible because of all the technical achievements. I'm not a specialist of the electron microscopy, but I think there was a lot of sample preparation. Even you have now more and more advanced techniques for microscopy imaging and automated picking of the images, and also some good statistical software for 2D classification, 3D classification, and 3D reconstruction or refinement, and then finally the fitting. I think what we will be presenting here, it will be for the fitting, having modeling good structures of the macromolecules. still there are many challenging things to be done in this last step. Sure, there are many difficulties for fitting. Now, with electron microscopy, we can see different conformations in the images, which have large changes between them. Then we have to be able to find the structure of the different conformation, different structure seen in the images, which are large changes. Then we have to be able to simulate them and to find their structures. And even for a given structure, you can have large heterogeneity, inflexibility. Some parts are more flexible than others, and we have to be able to model them. And then after the electron microscopy density maps, even if it is with medium range, let's say more than 5 Armstrong or 6 Armstrong, we have to be able to model. Still, there are many difficulties, but we have to find a large number of structures which have to be of good quality, with good energies. But the electron density maps with very high resolution also is very challenging, because we have to find a unique or two structure of very, very high quality structure, which is sometimes very difficult to find such structures because we have to fit exactly the density volumes are very, very precise. Then we have to have good software, good methods to have very precise structures. And then beyond the EM, then we have, because this is an opportunity that electron microscopy offers us, that we can see different conformations. And then we have to be able to see how we can go from one conformation to the other. What are the pathways? And there's also a good opportunities to test the methods and to find the dynamics of the structure, to see how they function, what are the precise dynamics. And this is a good opportunity that the X-ray crystallography didn't offer to us. Okay, now there are different techniques that are applied for 3D fitting. I think the historical ones which is widely used now are the rigid body fitting. I think different parts of the macromolecules are divided in different parts, and they are fitted separately. And I think the first work have been done by Baker and Johnson in 1996, modeling the FAB. But doing this is good method, but you don't have the flexibility between this rigid part, even you don't have the internal flexibility. Then for this, you need other methods, give you the flexibility of the molecule, like energy minimization, Monte Carlo, molecular dynamics. All these methods have been used in electron microscopy. And then you have more flexibility and better fitting. But the problem with this method is that you cannot deal for the real large changes of the macromolecules, like collective changes, global changes, internal changes. And there are some articles that treated with PCA. You can have the principal component analysis method, that the first principal components give you the collective motions, and there's some in the literature, some researchers use these principal components. But the other method is the normal modes. Normal modes, you don't need molecular dynamics to have them. With a single structure, you can compute the normal modes. And from then you have all these collective motions. And then there are many articles using normal modes now for fitting. I think I put here one which comes at 2004 by Tama. It was a student of my student. And he did this with Charlie Brooks. She was in California there at this time. then you see that in red is the crystal structure, and you have the electron EM map here, which didn't fit. Then they use the low frequency modes in order to change this conformation that you can get a good fitting. But they have to use normal mode plus energy minimization, because when you displace the structure along the mode, you have some structural distortion. Then you have to use some energy minimization to relieve distortion. But still, this is good, but you don't have all the flexibility. Then you need also to use some molecular dynamics. Then what we did with Mauricio Costa was to develop a method which combines molecular dynamics with normal modes in order to have even more flexibility, and then this is better for fitting. Then how to combine MDeNM and molecular dynamics? We have all these small and medium range fluctuations. You can explore a tremendously large number of energy minima. We have the full environment, but and the other side we have the normal modes, which have directed the large conformational changes, at least the directions of changes. But you use simplified potential energy, potential surface, and then and everything is done in vacuum. Then the best is to take the best of each side and to couple them, is why we call this MDeNM. MDE in Portuguese is N, MDeNM, but E is excited, MD with excited normal mode. That is, we are exciting the normal mode large collective motion by putting some kinetic energy and doing molecular dynamics. Are promoting the large changes and doing molecular dynamics within a molecular dynamics simulation framework. And then we showed this, and it was in 2015. It was done in the PhD work with Mauricio Costa. And we showed that we have a better and a more complete description of the energetics and dynamics of large molecular systems. Now in 2020, we just extended this method to implement with EMfit, electron microscopy fitting. And then this work was published in 2020. And this the focus, we will be focusing in this lecture, the use of this MDeNM-EMfit. Okay. Then I think, I don't know if in between the people who are attending, maybe some are not familiar with normal mode, then I want to do some reminder of this, then the thing will be more clearer for those who doesn't know the normal modes. And normal modes are, I think, was done at the same time as molecular dynamic simulation. It was in 1980s, like Paulo Bish was telling before. It was in the pioneering period in Martin Karplus laboratory, and I was there at this period. And then I worked a lot on the normal mode technique. And then we introduced this in the CHARM program. It was one of the very first programs in the early 1980s. Then everyone knows now that we resolved the Newton equation to have, then with molecular dynamics simulation, we have the numerical simulation, which is exact solution. And but normal modes consider only is based on analytical solution, not numerical solution, is approximate, and is due to a quadratic approximation of the potential energy surface. Then you get directly the direction of motion, this collective direction of motion, which are obtained by diagonalizing a Hessian matrix, which is which every component of this matrix is the second derivative of the potential energy function with respect to the coordinate, but is quite approximate. But what we observed is that it was back in 1990, even in the late 80s. I think even when I was in the Harvard, when we did these normal mode vectors, we were very astonished to see how the lowest frequency motions were describing the large changes of the molecule, even that we get some movies of them. I don't know where are the movies now, but it was we were very astonished. And now it is many people are working with normal modes to see these large motions. And many articles have shown that the large motions correspond to functional, most of them correspond to functional motions of the macromolecules of proteins. Then just to give you a picture of this is glucosamine 6-phosphate synthase, GLMS, what I was working, I think more than 10, 15 years ago. These white arrows show how the domains are moving. And this is a complex of two proteins. You see the motion here. And this one of the lowest frequency motions, but we have many different lowest frequency motions that can be used in the EM, that you can monitor this motion to fit the EM map. At the right, you have the CDK2 cyclin A, and you see how the cyclin A is moving all around the CDK2. And this corresponds to large, at least for glucosamine 6-phosphate synthase, it corresponds to large time scale. It goes over microsecond, I don't know, 10 or 100 microseconds. And it is impossible to use the molecular dynamics. We tried it when I was working with Nicolas Floquet, which was in Paris, and it was impossible with molecular dynamics to study these motions. Then the normal mode gives us a big opportunity to monitor these motions, and is very useful for EM modeling. Then it was in 1993 that we published how to diagonalize this matrix using all atom physical force field in CHARM. We implemented in CHARM. You can go up in a desk computer, you can go up to 50,000 atoms, but it becomes very computational, very intensive simulations, but up to 20,000 is okay, but it's not fitted for EM, for working with EM, for modeling with EM. Then we prefer now to use the elastic network model. This is called ENM, elastic network model, where they only consider C-alpha atom mostly, but you can have also all atoms, but it will be too much atom. But generally they use only C-alpha atoms, and between two C-alpha atoms up to a given distance between them, we put some strings. Then this is a whole network of springs. Then this matrix, the Hessian matrix defined by only the C-alpha atom, is 2 orders of magnitude lower dimensions for the Hessian matrix, and they can be diagonalized very rapidly, and is very suited. And we use this ENM method in the MDeNM-EMfit script that we will be explaining later on. And then what is astonishing, but I couldn't believe, it was after we published all atom physical force in 93, in 95, but after that, this ENM method appeared, and I was very astonished to see that the 3 or 4 lowest frequency modes which describe this collective motion were very similar to the one that we were obtaining with the exact physical force field. I would say at 70% overlap between them, which is quite acceptable and is very nice to work with them to gain CPU time, at least for EM mapping. Okay, if I take the vectors, all these low frequency vectors, Q1, the normal mode Q1, Q2, Q3, we can define a whole space. I put here only 3 modes to define the space, but you can get 4, 5, 6, and you have but we can't represent here in 3D. And then each point in this, I will say that this space is a normal mode space, that every point in the space corresponds to a given conformation of your molecule. These are large changes, the space of the large changes of the macromolecules. And then it's very easy to generate all this point. It's just a linear combination, displacements along a combination of the normal modes. Then you can generate this point very, very rapidly, but still you have some distortions when you displace because it's a linear displacement, linear structure displacement. Then you have to relax, then you have to do energy minimization or molecular dynamics to relax the structure and to get energetically sound structures. Then even that you can do some molecular dynamics from these red points and feel the whole cube, the whole space, normal mode space. Then one advantage of doing this, then you have an overall view of all the variety of structures of your macromolecule, and it's very interesting. But this is very cumbersome, that generate points and then do the dynamics separately for all the points. But it's feasible that we are trying this, but we find, but another way to go very fast is to simulate at the same time, generate this point at the same time as you are doing the molecular dynamic simulation. Is why the strategy that we did it with Mauricio Costa many years ago, is just to put the initial structure at the center of the cube of this normal mode space and generate the vectors which points at the different regions of this volume and every vector here corresponds to a linear combination of the basis normal mode vectors. And then we can disperse these vectors in an isotropic way. Then we don't introduce any bias. Then every region are on the same strand. And then we can start to do some molecular dynamics of the initial structure and just putting velocities which is along this vector. We are pushing the structure along this vector by molecular dynamic simulation. Putting velocity means that we are putting some kinetic energy to the structure, and this structure can evolve along this vector, but it doesn't follow exactly the vector. Then it can deviate because some region may be forbidden energetically or difficult to go there, and that they are forbidden region like in the Machandran plot. And then, but we can generate, and every time we are injecting periodically this velocity, then we have every delta t time, we renew the velocity injection. Then we just like a hammer, we are pushing the farther away the structure. Delta t here means that this is an excitation time, or we call it also relaxation time. This time allows to the system to relax because you have motor dynamic simulation, you have local fluctuation. Then the system will couple with these large changes, which is more rapid than the local change, and then you have to couple them and relax the structures. And then we can push it at every direction, and we can do the same thing along each direction in different CPUs, in different simulation. Then we can fill up all this volume very, very rapidly. And then after, if you can extend, you can do supplementary macrodynamic simulation in order to fill all this cube. And then we get very nice results. We have an overall view of the potential energy surplus, even that we were very close to free energy landscape that you can get from more sophisticated methods. Concerning the EM fitting, this will be very lengthy because your structure may be in one region of this cube, and then we have in this MDeNM-EMfit we don't generate all the points, but we generate only in a region where we will have the larger correlation with the experimental EM map. Then we will have a filtering procedure to select only the vector which will be pointing to the target structure, then discarding all the other vectors, and this will save us a lot of simulation time. Then we have many scripts which now many people are using. We have a script, MDeNM script, which is read by the CHARM simulation program. And then this was the extended to EM-fit script that is also read by CHARM. These are scripts that are available upon request, and we distribute in the scientific community. And the third script that was written by Mauricio Costa, it was called MD-Exciter, it was not yet included, but it will be in the near future be included in the Bio 3D module of the R package. Then it's very versatile, and for people who are not accustomed with the CHARM program, they will be able to use with NAMD, Amber, GROMAC, and maybe there will be other software too. I wanted just to show you how powerful is this MDeNM method. Just is one slide showing this is we take the example of the protease of HIV, where there are two spin label residues here that experimentalists using electronic paramagnetic resonance method, they can measure the distance between the two labeled residues, and they obtain an flap opening distances, which goes from 18 to 48 Armstrong, which is in red. And our method, using only 10 nanosecond simulation, we fit it very well to the EPR results. We were very happy with that. And we are also, when we did multidynamic simulation, even 200 nanosecond multidynamic simulation were far to be acceptable. We were only exploring from 24 to 36. Is 200 nanosecond, then you will need more than 1 microsecond, maybe even over than 1 microsecond to be able to describe all these distance variations. Okay, this is just to show you that this technique is quite powerful. And it was used by many researchers now. There were many articles published, and many work are ongoing also now for studying conformational transitions, studying allosteric properties, working allosteric properties, and even very interesting in this pandemic area to see new sites and do virtual screening, find new compounds. There a whole project to study the association, complexation of proteins, which one of them could have a large changes, even the dissociation. And a work that we have done also, we were able to see the helix strand transition. It was done with to see in the prion molecule with Anna Liga-Scott. We did an article, and by exciting the medium range, we not only the lowest frequencies, but also the medium range frequencies, which promoted these changes from helix to strand. Then we didn't see this with, it was very difficult with molecular dynamics and also impossible with normal mode. When you do the two methods separately, you don't get, but when you associate the both methods, we were able to see these changes. And it could be very interesting for the EM fitting, because we can also excite higher frequencies than the lowest frequency, just to fit small regions of the molecule, which could be very interesting. There's some work going on to study the enzymatic activity, and there's a work we published this year with membrane protein. The KIR protein is potassium channel protein that Charlene Pagnan will present you after Mauricio Costa presentation, and she will show you the motion proteins and also in relation the work which are being done in relation with electron microscopy. Then we will also work for interpreting the experimental size data, the small angular scattering data, to interpret a structural interpretation of what the experimentalist sees, because they don't see any structures. Then we can interpret this data. And finally, this year we were doing the work on the modeling in relation with cryo-electron microscopy. And then I will now leave my presentation and let Mauricio Costa present the technique that we developed for MDeNM-EMfit. Then I let Costa have his presentation now. Then I thank you. Then I did a general presentation. Now you'll see something more focused on the methodology itself. Okay. Then I don't know how to.
I guess I have to share my screen now.
Okay.
Yes,
I will take this away. I don't know how to do this. Okay. Okay. Now, Mauricio, you can present the work.
I'm sorry, David, I'm not sure, but I think we have a question.
I don't know if the question is now or maybe you can ask question after every talk. Okay, is a good idea.
So Eric, if it's okay for you, you can ask later your question.
Yes, I think that people can ask after.
Okay, that's good.
Okay, as you wish.
So I hope you can hear me. I want to thank the organization and Davi, Paulo Bicho for the opportunity to present this short talk. And the idea, as David introduced the overall principles of the method, now we are going to focus in the specific case where we use MD molecular dynamics with excited normal modes to fit atomic structures in Cryo-EM maps. This is a recent paper. It's a preliminary study that we published recently. And the purpose of this method is that we usually, when we are solving the structure of a given protein or a biological molecule by cryo-EM, we obtain a EM map in a given conformation. But sometimes we don't have a given structure in the same conformation to fit in this map. And sometimes we cannot solve directly the structure because we don't have enough resolution in the EM map. So we have to fit a given structure. Here I'm telling you that is in A conformation, for example, and the map is in the B conformation. This structure can be an experimental structure that you can find in the PDB, for example. If you don't have, you can generate a theoretical model. And sometimes, as David said before, the rigid body methods that are quite fast, they don't work very well, and we need some flexibility. This is why we developed our method, try to fit these structures when the rigid body methods doesn't work very well. So when we compare in the general case, the general MDeNM, the standard version of the method, and in this new version, while in the standard version we intended to explore the conformational space, try to be the more uniform as possible. Here, the idea is to perform a focused exploration, which means that we have a target state. We want to generate structures that are closer, each time more close to the target state. So this philosophy is quite different because it's a very biased procedure, because we are going in specific directions instead of exploring the whole conformational space. So the idea is to save calculations and to try to approach as fast as possible from the correct solution. And what is interesting, we can obtain several trajectories where we can have a lot of information about the intermediate structures that were generated in the conformational search, trying to approach the target structure. Here is the flowchart of the procedure. I will not go over the details, but the idea is to show you that it's not that complicated. It's not very different, in fact, of the original procedure. The only step that we have now, the extra step, is that we generate theoretical EM maps from the intermediate structures, from the initial structure also, and we compare these maps to the target map, which means that we can have a measure that's called cross-correlation, and all the time we are measuring how is the correlation between the intermediate structures and the target map. And the idea is that we are going along directions of the normal modes that increase the correlations with the target map. I will now present you the first example that where we applied this method, and we took a very known protein that is called the lysine arginine ornithine binding plasmic protein. It's called LAO. And this protein we have some structures in the PDB, and they are found in the open state, shown here in blue, and in the closed state in red. And when we analyze the conformational space, when we project these structures into the directions of the principal components extracted from this set of structures, we can see two groups: a group of open structures and a group of closed structures, almost They are very similar. And we took a given one from the open group, the 1PDB, as our initial structure, and the target state a closed one. Instead of here in this first application, instead of using a real EM map, we generated the synthetic maps from the target state. For example, here we worked in 3 different resolutions. We worked at 3 angstrom, 5 angstrom, and 8 angstroms. You can see the difference in the level of detail when we changed the resolution. And that being said, when we work with a real EM map, we are usually working around this range of resolution. So we try to test our method in a range of resolutions that are commonly present in the generated EM maps nowadays. Here is an example of the trajectory of the solution. It's important to say that we don't have only a single solution. We have an ensemble of solutions, which means that we may have several different solutions, but here I will show you only one. And again, in red, we have the target map. Here's the synthetic map generated from the initial structure, and I will try to show you the trajectory here. As you can see, from each intermediate structure, I was able to generate EM map, simulated map. And I was always comparing each of these maps with the target one and the correlation and the cross correlations. And I obtained the best solution here that you can see that is quite similar to the target map. In this case, as is not, I had the structure, the target structure. I could also compare directly the structures, and as you can see here, the starting one is in blue, and it will move almost perfectly towards the target structure. And in this method, we were able to perform, I guess, about 30 or 50 trajectories. Some of them we obtained best solutions. Here I'm showing you one example at 5 angstrom EM resolution. And when we compare the results, our method was able to correctly obtain a solution at 3, 5, or 8 angstroms. The RMSD between the best solution and the target structure was very low, at approximately 1 angstrom, which means that the structures are almost, it's a very nice match between the structures. And as I said before, what is interesting about this methodology is that we can also try to understand how the structures move from one state to the other, which is quite interesting because we can obtain some interpretations about the energy barriers separating these conformational states. We can try to understand why there is a separation between these conformational states. So we can try to understand also the energetics of the conformational transitions. When this is the most interesting aspect of the method, because we have, as David said before, we have normal mode based methods. We have also other methods that are based on molecular dynamics. And why our method is, what is the advantage of using, to combine them in this context, is the structural quality of the solutions. Because when you use normal modes only, you displace the structures, it's quite often you can deform the bond angles or even the dihedrals. You can have superpositions between van der Waals spheres, so you have structures that are not sometimes, most of the times, they are not physically you cannot, they are not correct. So our method, as we perform the simulations using all atom force field, in our case we used the charm force field, and the whole exploration, we can also add water molecules. We can add several layers of complexity to the simulations, which means that the solutions that we obtain, they are always physically accepted. Here we compare the number of clashes. The clashes is defined by the superposition of the van der Waals spheres between two atoms, and we compared our solutions with those found in the target state, and we obtain even fewer clashes if compared to the crystal structure, which means that our structures are, according to this parameter, they are very nice. And we also compared the score that mole probability yields. That is a combination of several metrics, and the values obtained were in the same range that we obtain for the target structure, that is crystal structures at 2 angstrom resolution, which means that our solutions are all acceptable in terms of the quality of the structure, which is a very nice point, because you can use these structures later for docking study, for molecular dynamics, which means that you don't have to correct the solutions after the application of the method. They are ready to be used for another purpose. So as I'm trying to summarize all the advantages of the method and why should we use it. And we have the accuracy, because as I said before, we can obtain solutions that are very close to the target structures. Of course, we are now applying the method to a whole set of examples, because in this first publication, it was like a proof of concept, but now we are extending a data set of proteins, because we wanted to define what are the good parameters, and to make it work in very different molecular systems. But the great advantage that MDeNM-EMfit offer is the obtaining of high quality structures while doing fast calculations, because these calculations can be, now in this first version of the program we used CPU, but it's adapted for computational clusters. But it will be easier, it will be very easy to adapt the future version of the program to the use of GPU calculations using the molecular simulation program that like NIMD or GROMACS. So we have we have a lot of room for improvements. And another important point is that we can use this method to any type of protein. It could be a soluble or membrane protein, even a theoretical model, because as David said before, you only need the structure for calculating the normal modes, as well as for perform molecular dynamics. So we don't need a lot of information to start the method. Of course, if you have additional experimental information, it's always useful, but we don't need it's not required. So I hope I was able to explain to you the principles and how the method works. And now I will leave the presentation to to Charlene Fagden, and she will present an application, a future application of the method, where we are obtaining, trying to obtain the EM structure, the EM structure, the cryo-EM structure of the KIR protein. And of course, we will have to use flexible fitting algorithm with MDeNM to obtain the right high resolution structure at the end of the procedure. So thank you again, and I'll now stop sharing my screen.
Okay, thank you, Mauricio. I will share my screen. Do you see my presentation?
Yes, we see.
Okay, thank you very much. So first of all, thank you to let me the opportunity to present my work here. So as Mauricio said, I will present in first part the application of MDeNM method presented by Dr. Perahia, and in the second part the future application of Cryo-EM-EMfit method. So to do this, we work on a KIR-BAC 3.1 channel, and I will shortly present it to you. So it is a transmembrane protein that is ubiquitous, as you can see here on this schema. It is responsible for the transport of potassium ions, and it creates and controls the transmembrane actin potential. It is involved in a biological process like the creation and propagation of the neuronal action potential, the regulation of the cellular volume, muscle constriction, and heart pulse. Here you have a description of these channels. It is a KIR-BAC 3.1 homologous bacterial, and it is composed of 3 domains. At the bottom, the intracellular domain, the cytoplasmic domain, and in the middle, the transmembrane domain, and at the top, the extracellular domain. So we use MDeNM method to understand how does this channel open and close at the level of the transmembrane domain, and the impact of mutation of this opening. And it is important to understand that because some mutation of this channel can cause the syndrome, the Andersen syndrome, a rare disease. So MDeNM, thanks to MDeNM, we obtain population of MDeNM structure in function of the state of the channel. So we have the open state that represented 7% of the total structure, closed state, the half of the structure, and intermediate state. And we compare this result with electrophysiology studies led by Dr. Saïd Bendaoui in Nice, in France, and it found an open probability of 9.9%. That is very close to our 7%. The difference can be explained by our definition of an open state in MDeNM, because we were very strict on the criteria of opening to be sure that the open structure were really open. Here you can see some sample of the structure from MDeNM, from very closed structure, where water can pass through and potassium not anymore. But here in the open states, they can go through with 2 intermediate states. But MDeNM can give indication about global motion. Like here in the transmembrane helices, we see kink motion of a transmembrane helices that is involved in the gating of a channel. It was seen in experimental data, like in 2D crystal, where in a closed state we can observe a kink in the transmembrane helices, and here in open state there is no kink. And in the cytoplasmic domain, we observe two motion, swing of the cytoplasmic domain, found in atomic force microscopy 2, and a rotation of a cytoplasmic domain. And it is interesting to know that the two structures in open and closed state on the PDB, reserved in crystallography, has a difference of 30 degrees at the level of rotation of cytoplasmic domain. So, and in the last part, we compare the MDeNM result with experimental data realized by Dr. Eric Forest at Grenoble in France. It was a study of hydrogen deuterium exchange using mass spectrometry. Here you can see the protein, and when it is in red, the residues are very flexible. When it is in blue, the residues are very rigid. So at the bottom of the transmembrane helices, you can see it is red, so the residues are very flexible. That is correlate with the kink of the transmembrane helices. And here you have a loop, and it is the highest, it's representing the highest, the residue with the highest flexibility. And this loop has the residue involved in the swing of the cytoplasmic domain. So MDeNM obtained results in agreement with experimental data. You can see more details in this article. But MDeNM gave some clues in global motion, but when we study a mutation, we could see local motion of a protein. For example, on the left, you have a wild type, and on the right, you have a mutation. And the mutation caused an increase of opening of a channel. And we understood that is because of P interaction, and so a local motion. So in the second part, in the first part, I presented the work on KIRBAC 3.1, but our goal is to work on KIR 2.1 because of the disease. And Professor Catherine Veillon-Brian, my thesis director with Dr. Perahia, took some picture in Cryo-EM at Pasteur Institute in Paris. And here you can see one of the beautiful pictures she took recently. And here in blue circle, you can see the protein, so KIR 2.1 protein. And we manually picked this particle on all the image took. Then we classify all the particles in function of their of what they look. For example, so similar particles were classes together. So it gave a 2D classification. At the beginning, we had 37,000 particles, and we eliminate classes that don't correspond on the projection to the projection of the model of KIR 2.1. So it remains 17,000 particles to build in 3D the model. But to do that, we need an initial model. So with homology modeling, we model KIR 2.1, and we low-pass filtered at 40 angstrom to obtain a neutral-link density. And we compute the 3D map of KIR 2.1. We obtain a map at 6.2 angstrom. And here you can see the result. And we can see some features like the outer helices here and slide helice here. So the next step and the final step is the fitting, like Dr. Costa said before. So the fitting is you have your map and you have a model, atomic model, and you try to match your atomic model into the map to obtain something like that. To do that, we first execute a rigid body fitting with one chain in KIR 2.1 in the map, experimental map. But as you can see, it is not perfect. We have some problem, for example, here, because it is rigid. We can't move the backbone or side chain. So our plan is to use the method described by Dr. Costa and the MDeNM-EMfit, because it is a flexible fitting. It concerns all atoms, so the backbone and the side chain, and it permits you to reach a more accurate atomic model to fit into the electronic density. And it is faster because it is not a manual fitting, but the complexity of this project is very high because, for example, the previous example given by Dr. Costa concerned theoretical data, theoretical map, but here it is an experimental data, and the system is bigger. There is many atoms, and it's separated in 4 chains. So it will a challenge to fit it in the map. So thank you for your attention, and I will let Dr. Perahia conclude this talk. I cancel the talk.
Okay.
Thank you.
I will conclude.
You will talk something?
Then I just wanted to acknowledge I have to go through all this. Okay.
Sorry.
Just I want to just, I wanted to acknowledge all the persons. Where is it? I wanted to acknowledge all the persons who worked, who collaborated, either that with MDeNM, they applied MDeNM for many different proteins, like in Brazil, I had many collaboration with many persons, many researchers who applied MDeNM, first in the Institute of Paulo Bish and Pedro Pascuti, and still in Oswaldo Cruz Foundation. Yeah, Mauricio Costa is from Oswaldo, but we worked a lot with Paulo Batista. Giselle Rocha did a stay, in 1 year stay in our lab. We applied the MDeNM and write a new program with MDeNM to find pathways to go from one structure to a target, but not based on the EM. In University of UFABC, I collaborated a lot with Scott, Anna Scott, Antonio Braz, Pedro Tulio. Pedro Tulio is now in Paris with me. We are working on new methods, which is more normal mode is adaptive normal modes, which are working very hard on that. And UNESCO, I have a lot of collaboration with UNESCO in Botucatu, with Angelo Magro, who spent many times, he came many times and stayed some time in our laboratory. And also with Marcus Fontes in Botucatu, we had large collaborations. Gomez, Antonia Gomez, he is now in Montpellier, but we are collaborating with him. Carlos Fernandez, now he will be coming in January to the Professor Venn-Embryon laboratory, and I think we will continue to collaborate. And in France, all the people in the Professor Venn-Embryon laboratory who were studying for the in the experimental part for the young. In l'Institut de Biomolécules Max Mousseron, they had Nicolas Fouquet, which was also applying MDeNM with my caproscopic program with Paulo Bisch and Nicolas Fouquet, and Pedro Renaud, who was in Rio with Paulo Bisch. He was in Nicolas Fouquet's laboratory, and now he is in Spain. And there are other people like in Said Pandau who did all these experiments, electrophysiological experiments. Here I put twice. And the researcher in the Institut de Biologie Structurale did also the HDX-MS experiments. And thank you very much for all your attention. And maybe we can now answer your questions. Maybe it's quite late, but it took more time than were long. But okay.
Thank you very much, David. I think that Leandro has a much more ability to announce questions than I. So I will just see what Leandro will do.
Sure, sure.
Is okay, Leandro?
Yeah, sure, it's fine. First of all, David, thank you very much for this very, very nice talk. We have some questions here. I made some notes here regarding the questions. First of all, according to my notes, is Eric Allison. Eric, are you there? Even he has he raised his hand. Eric, I think you are allowed to open your mic. Is that okay for you? Just wait to see if Eric, are you able to listen to us? Yeah, I'm not sure, but okay, we can wait for a while. Also, Fernando de Oliveira has also a question. Fernando, are you there? I'm not sure. Let me see if I find Fernando here. Fernando, can you open your mic, please?
He's saying that he's trying to turn on the mic.
Hi, can you listen to me?
Yeah, but who is talking?
Loud and clear.
Hello,
Hi, Fernando.
Hello.
Hello.
Nice to see you again, Professor Perahia.
Nice to see you too.
So I have two main questions. The first one is regarding the MDeNM-EMfit. When you start the process,
what is your question?
Fernando? I think Fernando, are you listening to us? We are not listening to you any longer.
Yeah, I'm listening now.
Yeah, please, if you can ask the question again. I'm sorry.
Yeah, sorry. Regarding the initialization of the MDeNM-EMfit protocol, what are the initial coordinates do you use to initialize the process?
It is the open X-ray structure.
But in a real scenario, when you come with a electron density map from a cryo-EM, do you initialize it from the open? Sorry for the name of the tool again, please. I couldn't hear.
Say again.
You initialize from a Cryo-EM map from this tool you mentioned before?
We did an artificial EM map for the closed state, and we took the open state as the initial starting structure, whose theoretical map didn't correspond sure to the target map. And then we tried to test the method how we can converge to this map. But
okay, thank you. This answers the second question as well.
Yeah.
And I try to answer the first one. I think I got it. When you have a real case where you have only the EM map, you have to search for a structure of your system in the PDB in another conformation. If you have in a conformation that's quite close to the map, even better. But sometimes you can find in the PDB or the protein in a very different conformation. Or if you don't find in the PDB, you can generate a model and apply the procedure using the model as the starting structure.
It's like using a gathered constraints for homology modeling. In Modeler, you generate constraints from homology, and the procedure is similar, as you mentioned, right? You get an initial structure from the PDB by homology, and then you relax it using the EMfit, right?
Yes.
Yeah.
Okay.
But you can either create by homology. If you don't know the structure, you create an homologous structure and you transform it with MDeNM.
Yeah. Okay. Thank you very much, and thank you for the presentation. Nice work.
Thanks.
Thank you very much, Fernando, for your question. Let me try again. Eric, are you around? Can you listen to us? Eric Ellison. I'm not sure. Okay, not yet. So also Junko Tsukamoto. Junko, are you there? Junko Tsukamoto. You can also turn on your mic. Junko, are you listening to me? No, he's not here.
I think there was a question from Alison Julie.
Yeah, this is Eric. It's Eric Allison, I think his name.
Okay. I
can make the question, but I would like him to speak if he's able to.
I can read the question. Thank you for your thought.
I put an answer for it, for a written answer also, but I didn't know that he was online or not. I tried to answer.
Yeah, can you apply your method to determine which is most visited conformation for a complex when you compare with different states determined by Cryo-EM? I think yes, you can determine, because MDeNM give you the distribution of the structures. Then you can see what are the regions the most populated, and you can have an estimation which regions are more visited. I don't know.
Okay. I think the last two, we have another question from Pedro Tulio. Are you there, Pedro? Are you listening to us?
Pedro Tulio.
Pedro Tulio, yeah. Let me check.
Hello.
Yeah. Hello, Pedro. How are you?
I'm fine, thanks.
Hello, Pedro.
We are fine also, thanks. You may talk.
Thank you very much for the lecture. Nice work to everyone. And I have a question to Mauricio. I found very interesting the plot that he showed to us, comparing the trajectory of the three different resolution during the process of solving the theoretical map. That the biggest resolution took a very different path from the other two, and but in the end the residue was very similar to the other. So what I'm trying to ask is that did you had the time to analyze how this conversion took place? Because it's very interesting to see maybe some barrier that the lowest resolution it's easier to surpass during the conformational change, and the biggest resolution was not able to this path, and then other motions were combined to arrive at the final structure.
Well, thanks for the question. Yes, the point is, in this plot, I was showing the trajectory of the best solution, but we actually have, if I remember correctly, I performed about 30 simulations for each EM resolution. So instead of having only one trajectory, we have like 30 trajectories. And not all of them arrive perfectly at the target state. And instead of analyzing only one trajectory, I have to analyze the whole. If I am interested in understand about the transition or the energy barriers, I have to analyze an ensemble, a lot of trajectories, and trying to find the average properties from these trajectories, because I don't even know if the number of trajectories that we generated was it was small, because for this first publication it was for a special issue. We didn't have the time to perform a lot of simulations, so we had to pick a very small system and perform a reasonable number of simulations. But for now, the idea is to apply more a higher number of trajectories.
Possible to do it.
Yes, now we have the time.
Number of trajectories and simulation.
Yes, because
very interesting this path property. I found very beautiful in the presentation.
Thank you.
I think there are many rooms for further developments to increase the efficiency of the method.
Yeah.
No, thank you very much, Pedro. Do we have any other question, or someone that would like to make some kind of comment or something like this? Junko mentioned that he has some problem with his microphone. I also asked him if he would like to write his question. We can make it, but I'm not sure he is able to write the question right now. So I don't know, Paulo, if you have, do you have something to mention or to comment?
Just that it was very nice to have all these people talk about some very important issue now for the electron microscopy. Someone is so talk else, but the most challenging question is how to fit this electron density maps to the usual representation in the molecular dynamics of atoms. I have not understand well. We create boundaries to put inside the molecule, or what you do exactly? Maybe or Mauricio.
I think we generate, for a given structure, we generate the overall density map, and we compare only the density maps with the experimental density maps. And we have many tools doing that. You can have two density maps. You can superimpose them and see a correlation between them.
Okay, so there is a step more that means that you must create at each step.
You have to recompute the density map and compare.
Okay.
But it is done in unbiased way, because some software tries to put a penalty function directly in the potential energy function. Here it is not. We generate and see what are the structure which fits well, and we don't. But we have less bias than other, but putting directly the penalty function.
Okay. So thank you. I don't know, Leandro, if there are some other questions.
I'm not sure if you'd like someone else would like to make something. I see that Pedro Pascucci is around, that you mentioned him during your speech. I'm not sure if he's able to talk, because first of all, you write to him. I'm not sure you or someone who write to him. But...
I wanted to say.
Hi, Ray.
I want to just say congratulations, David, and the cool work.
Hello, Pedro.
And very nice work.
There is also Paulo Batista, maybe he can speak some.
Can you hear me?
Hello, Paulo.
Yeah, we can.
Okay, very nice talk. It's very good to see all of you in this meeting. And I was wondering and how to apply this EMfit method when you have a more complex system, for example, a large complex with hundreds or dozens of proteins, and how to obtain a first model to try to fit.
It's a good question. But I think doing by part first, I think is something by parts, fitting some, not all at the same time, but taking few parts and doing step by step, I think. But this is a challenging question, challenging things to do. Yeah. But
and also a more technical question about the orientation of the because to perform the cross correlation, to calculate the cross correlation, you can have many different orientations of the molecule. So do you turn the molecule to see which is the best position?
But I think they are superposition program, like in Chimera. Yeah, they superimpose two density maps, and they do all this superposition.
Yeah. So first you need to have those superposed to the.
Yes, you have to
superimpose the electron density.
Every time you have to superimpose, and then you can superimpose some sections separately and see how well it fits. Then superimpose other parts. If it is very complicated, multi-protein complex, then I don't think it could be done one step, but we have to. I think we have to take many steps, many fitting steps of different parts, and then try to integrate all this. Yes.
For example, by
But these are
segmentation of
challenging.
by the segmentation of the whole electron density, for example.
Yeah, probably. Yeah.
Okay, thank you very much. It's very nice just to see you, even remotely.
Nice to talk to you.
Congratulations for the organizer also.
Thanks.
Thank you also, Paulo. So do we have another comment or question?
I ask a little bit to David. I don't know, David, if Catherine want to speak something about this experience to interact with theoreticians. She is a very nice experimentalist, and I don't know she want to talk something about.
What is your? Yeah.
Hello.
Hi.
Hello, everybody. So yeah, I'm the experimental one here. So I do what I do. I express the membrane protein. I did and I work for a long time on the homologous of the human potassium channel, which is a Kirback, and now we are concentrating on the human potassium channel, which is a Kir, and it's much more difficult to express. So you have to be patient in order to produce it, to express it, to purify it, and then the taking image is quite a challenge as well, because here, I mean, the image Charlene showed, it took us quite a long time to get such a nice pictures. And of course, you need very high resolution, very high quality picture in order to get a good structure. But it's quite interesting to work with a theoretician, because you know, there are some questions that we can't answer just by the experiments, and working with people such as David and Maurizio, so we could explain some the dynamics of the protein, because using a cryo-EM, we just have some kind of snapshot of the conformation. But then you can, you know, using the MDeNM, we managed to explain how this channel open, because this channel is dynamics. And using this MDeNM method, we managed to understand how the getting molecular mechanism. So it's very complementary. Yeah, so it's a very complementary interaction between experimental and theoretician. Yeah.
Thank you. Leandro, I don't know if there are some more questions.
No, at least from my view here, we don't have any other question or comment. And if we don't, I'd like to ask you all to open your cameras, because we make our traditional picture to put on our social media. You can take out the glasses or put on the glasses. Please, everyone, and you can open your cameras, please. I don't know if Antonio would like to say a few words regarding our meetings or this meeting in particular.
Oh, me?
Yeah, you are the president.
No, I already said something at the beginning. I just want to thank everybody again for supporting our series of virtual talks. And it was very good to have you all here. This is the 7th talk we have this year. And I hope you guys take care. Pandemic's not over, not yet. We're almost there, but not yet. And I hope to see you all with us again next year in this new series of talks towards the IUPAB meeting in Foz do Iguaçu in October. Thank you so much. Yeah, take care. Now
thank you all.
Yeah. Thank you very much. Thank you very much.
So the picture, okay. Thank you, guys. Thank you a lot. thank you for your participation in not only today, but also in these few past months. Thank you, and see you around, and take care.
Thank you.
Thank you. Thank you very much.
Thank you very much.
Bye-bye.
Bye.
Yeah,
Thank you very
bye-bye.
much.
Bye-bye.
Bonsoir.